Hello everyone, and welcome to today's webinar, Accelerating Microscopy Image Analysis with AI. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Yung Choi, Ph.D., AVIA Product Manager, Product Owner, Leica Aivia. Yung, you may now begin your presentation.
Hello, my name is Yung Choi, and I'm the Product Manager, Product Owner for Leica's AVIA software. Thank you very much for joining me today for this webinar, where I'll be showing you how you can incorporate AI into your microscopy image analysis for accelerated insight generation. Today, I'll be showing you AVIA 11's key features focused on accessible artificial intelligence for scientists, which include whole experiment support from multi-well import, navigation, tagging of experimental conditions, generalist deep learning cell segmentation based on Cellpose for 2D, 3D cell segmentation for a wide range of cell types and microscopy modalities, workflows where you can build enhanced batch processors from history, spatial biology tools via Cytomap integration, where you can output AVIA's measurements for advanced analysis tools such as dimensionality reduction, machine learning-based data clustering, heat map, and hierarchical gating. In addition, we'll be talking about AVIAweb, which now includes permanent storage in the cloud. AI is becoming an increasingly larger part of our daily lives. You may have seen this in your own life, as in the case of a robotic waitstaff or self-driving cars that are growing in prominence. There are other examples where AI is deeply involved in your life in ways you may not be aware of, as in the case of robots in industrial applications, as you see here in Spot by Boston Dynamics, that explores dangerous underground conditions, or in medical settings where companies are racing to find AI diagnostic and therapeutic tools for various diseases and conditions. We also see this in the biological community embracing AI, as shown here by the exponential growth in the use of AI in the biological sciences. The number of publications with AI in biology or neuroscience in the last 20 years have grown over 360%. In the first half year of 2022, the publication numbers have already surpassed the total number of publications in the entire year in 2015. With the help of AI, scientists in various disciplines are increasingly getting productive and get to more accurate results faster. With more innovative deep learning models being introduced and getting refined, this trend will only increase such that we can anticipate researchers will likely be using a growing number of AI-assisted tools to further accelerate their path to groundbreaking discoveries. AVIA has been in the forefront of enabling biological discoveries fueled by AI-based image analysis. Built on the foundation of over 32 patents in machine learning and computer vision over the last 20 years, the AVIA team located here in the Seattle Leica Innovation Hub has created multiple innovative tools to help researchers worldwide to get to insights faster with greater accuracy. With AVIA 5, the AVIA team introduced neuron and spine analysis and machine learning object classifier, followed by deep learning model for EM images in AVIA 6, machine learning pixel classifier in AVIA 7, 3D cell tracking and lineage tracing in AVIA 8, deep image restoration, Python integration, and cloud-based AVIA for accessibility for all in AVIA 9. In 2021, we introduced AVIA that Learns, which is a deep learning-based object detection tool for easy-to-use AI without knowledge of coding or machine learning. With AVIA 10.5 introduced in February of 2022, we expanded AVIA that Learns for neuron analysis, where there's over 73% reduction in time needed to generate optimized parameters for neuron tracing assisted by AI. Using our built-in optional telemetry feature, users' data can help us to further refine our predictive model for continuous improvement. For AVIA 11 that we're launching today, we wanted to create tools that would increase productivity and efficiency from multiple different angles. First, we tackled the well-known challenges for cell segmentation, where there's often a trade-off for flexibility or automation. While many tools out there offer great level of flexibility, they might require full manual labeling and thus are time-consuming. On the other end of the spectrum, there may be fully automated tools based on deep neural networks with parameters estimated on large training datasets. However, these tools may be trained on specialized datasets and may not generalize well to other types of data. Cellpose, published in Nature Methods in 2020, is a generalist deep learning-based segmentation algorithm that can segment a wide range of images from cells, published by Carson Stringer et al. From Maria's Pakitariu lab at Janelia. In brief, this model uses two major innovations. First, a reversible transformation from training set masks to vector flows that can be predicted by a U-net-based neural network, and a large segmented data set of varied images ranging from biological to non-biological across different microscopy modalities. In this image, you're seeing a t-SNE output, which shows a wide variety of data sets used to build the model. To briefly describe the model architecture, in the training set, manually annotated mask sets were transformed via heat diffusion simulation to derive spatial gradients to generate flow representation as you see here. Then, a U-net-based neural network is trained to predict the flow field based on horizontal, vertical flow, and lastly, probability of pixels inside cells. The predictive flow fields are used to construct the predicted masks in the final step. For AVIA 11, we have integrated the 2D probability map generated by Cellpose into the existing AVIA workflow for ease to use deep learning cell segmentation. There are 4 new Cellpose-based tools that are created within AVIA. They are 3D cell analysis with Cellpose recipe, cell count Cellpose recipe, both of which output objects, Cellpose enhancement, image enhancement tool that outputs a confidence map, and lastly, Cellpose object detection image enhancement tool that outputs masks. Cellpose has been demonstrated to generate accurate cell segmentation for varied cell types for nucleus, membrane, and cytoplasmic stain for DIC and fluorescent images. Our benchmarking data show an average speed improvement of over 80% for this integrated image analysis pipeline compared to the Python interface to Cellpose, for which AVIA was the first commercial entity to do so in AVIA 8.8. So you're seeing this very significant speed improvement with the current iteration that's a fully integrated Cellpose into AVIA's pipeline. Here are some segmentation examples with AVIA with Cellpose, where this model can accurately segment various cell types for membrane, nucleus, and cytoplasmic stains. Here are some examples of the types of diverse image types Cellpose can handle. For example, biological images from cell image library and additional images from various microscopy techniques and object types, including fruits, vegetables, fish scales, and so on. Cellpose has been demonstrated to segment diverse cell types with higher accuracy than Stardust and Mask RCNN. Here's an example from a challenging data set where Cellpose can accurately segment membrane-stained cells. Then using AVIA's cell analysis recipe, the nucleus and vesicles can be segmented with spatial measurements such as vesicle distance from the nucleus or the membrane. This is a 3D image of intestine organoid courtesy of Dr. Andreas Mohr at ETH Zurich. The second feature we've implemented to increase productivity and efficiency is our newly built Experiment Explorer that enables multi-well image import. With this tool, we've enabled annotation of wells with different experimental conditions such that drug dosage or genotype of the cells can be tagged so that you can perform downstream advanced analysis. In addition, we've created a flexible, customizable workflow processor for enhanced batch processing that incorporates all the analysis tools available in AVIA, such as image enhancement functions, AI-powered pixel classifier, deep learning models, image analysis recipes, and object classifier into a single workflow. You can now analyze your image using AVIA's built-in tools on a single field of view and build an analysis pipeline using the new workflow tool. The analysis workflow you built can then be batch applied to your entire imaging experiment or just on select wells with your annotated conditions. Lastly, we've created a data export function for advanced data exploration. For this, I'll show you an example using Cytomap, a spatial analysis tool for cells, regions, and tissues, created by Caleb Stoltzfus et al. In Michael Gerner's lab at University of Washington. And Cytomap was published in Cell Reports in 2020. Cytomap enables cellular patterning, distance analysis, spatial correlation and dimensionality reduction techniques, among others. And AVIA users can now export out measurements per condition per analyzed objects from AVIA for advanced in-depth analysis. Here are some interesting examples you can achieve using Cytomap. On the left, you're seeing an example of UMAP dimensionality reduction that reduces multiple parameters down to 2 to allow for visualization of complex data sets and subsequent hierarchical gating of different population of cells for further analysis. On the right, you're seeing an example of Pearson correlation coefficient that visually demonstrates where meaningful correlations may lie for the measurements for different drug conditions for the cell objects that are analyzed in the AVIA in this example. There are various advanced tools available in Cytomap, and UMAP is one such example. UMAP is a nonlinear dimensionality reduction technique that allows users to create 2 new parameters from a high-dimensional dataset for visualization and analysis. You can see an example of this woolly mammoth in 3D that can be reduced using UMAP using different parameters. Using this, along with other advanced features in Cytomap, AVIA users can tackle complex datasets to generate insights faster. Our new Experiment Explorer can be used for images on slides such as this mouse coronal brain section for segmentation within AVIA and downstream analysis in Cytomap. Now we'll be moving on to AVIA for a demo of all the features. Now I'll be moving into AVIA to show you these features, starting with Cellpose. In this first example, I have the large intestine organoid sample that I showed you briefly within my presentation. It's a very nice 3D stack with 3 channels for membrane, vesicles, and the nucleus. In the interest of time, I'll be moving on to a cropped, smaller data set, which I'll be using to show you the workflow and show you the final output. In this small data set, as you can see, we see very clear membrane stain, and the nucleus stain actually isn't quite as ideal in this particular example, but as you will see shortly, Cellpose algorithm actually can handle both types of signals in this particular example. What I'm showing you here is within the recipe, we've introduced 2 new recipes. The first one is the 3D object analysis Cellpose that reads 3D data and outputs an object. The second one is cell count Cellpose that works for 2D images, outputs an object. In addition, we've introduced two new additional image enhancement tools that's available under the Image Enhancement Tool tab. The first is a Cellpose enhancement option that outputs a confidence map. The second option is a Cellpose object detection option that outputs a mask. In this example, I'm going to be choosing the Cellpose enhancement option to generate a confidence map for the membrane and for the nucleus, and feed them both into the existing 3D cell analysis recipe within AVIA. I'm changing my input to green and choosing the correct model in this case for the membrane is actually model number 3, which is based on Cytomap 2. Now, in this particular option, you do need to input the size of a typical object diameter. For this, I'm getting out of the 3D view to a 2D view, then choosing the annotation option to measure the diameter of a cell. This is roughly about 4 microns, so I'm going to input that value and apply to the current frame. So here's a confidence map that's been generated. I'm going to adjust the lookup table so you can see the objects better. To me, it looks like a really great confidence output. So I'm going to be using this into the recipe. Now I've actually run a similar process for the nucleus as well. So now moving into 3D cell analysis meshes recipe. So for the cell input, I'm choosing the newly created cytoplasm confidence map that you just saw. Then for the nucleus, I'm also inputting the nucleus confidence map. For the first step of whole cell detection, I'm going to be turning off all the other channels, the confidence map channels, so that I can see the output better, the preview output better. So this is an example of what the whole cell detection would look like. It shows the objects are segmented. And moving on to the next item, I'm going to actually detect nucleus in a similar way. You'll see these options all have different sliders that you could adjust cell diameter using the same method I just showed you with the annotation tool to get a size estimation, then enter as you need. We recommend for the Cellpose algorithm that you turn off all smoothing and background subtraction to start with and add on these options as you go. For the nucleus data, I'm actually choosing to remove background because my preliminary output showed that in this particular example, where as I mentioned to you, the nucleus data wasn't as well defined, having the background removal actually resulted in better segmentation. That looks pretty good. And now we're moving on to the vesicle detection step. So again, I'm turning on the preview channel. And it looks pretty good to me in terms of the placement of the vesicles. You do have the option to actually apply partition for the vesicles based on the size. And in this example, I've skipped background subtraction and image smoothing. Now, once I'm happy with the parameter, I run for the entire stack, and here's my output. I can adjust the color map here to, in case I'll choose color by name that shows you all the different cells in different colors. When you see the objects placed inside the membrane signal, you can see that the actual cells are pretty well segmented. For the vesicle, I'm going to actually color by measurement and choose, in this case, color by measurement, and choose measurement distance to nearest edge of nucleus. Let's see how that looks. And this is the color map we get. So this is essentially how you go through the Cellpose to AVIA recipe workflow. Now I will show you how it looks on the entire image. That could take several minutes depending on the size of the data. In this original image where I've applied the exact same steps I've shown you, on the entire image, you can see the detected objects. There's an example of the confidence map that I'm showing you right now. It looks very robust, doing a great job of segmenting already at the confidence map level. And I'm showing you here the detected objects for the membrane bound cytoplasm, as well as the nucleus and the vesicles. Here's another example of 3D organoid where we employed a similar strategy of generating a confidence map using Cellpose, then using the 3D cell analysis recipe within AVIA.
That was a brief introduction to Cellpose and how we use Cellpose within AVIA to get you more accurate cell segmentation. Now we'll move on to the second feature of AVIA 11, which is the new Experiment Explorer, which enables you to tag different conditions within different wells on a multi-well plate and enable you to do downstream advanced analytics via Cytomap. You can import your multi-well file formats in Leica live file format, or import a previously saved AVIA experiment file format directly into the Experiment Explorer. The way you navigate through this interface is quite intuitive. You click on the well, then you can see the number of fields of views there. In this example, you're seeing 4 fields of view per well. Right now, I'm showing you how you can tag different columns of wells into same experimental condition group, in this case, drug and no agonist. In this experiment, we actually had 2 experimental conditions. So now I'm tagging the second condition for columns 7 to 12 with the agonist condition. Now I'll select the first 2 wells in each condition to actually denote them to be my second condition, so that I can just run batch on these few wells in the interest of time. So I've done that. Now moving on to the data set. There are actually 3 channels in each image, but I'm going to be using only the first 2 channels for this demonstration. As you can see here, you can see quite a number of punctate objects. This is an experiment where the effect of agonist on the number of punctate objects will be measured. Then I'm actually changing the input channel as the green channel. Then I'm actually changing the color of the want pixel class into pink so that you can see it better. So the way the pixel classifier works is you train the software with a simple stroke. In this case, I'm actually training the software to recognize background, and I'm actually including some of my signal in this particular example. And the reason for that is to capture the background area around my want signal as closely as possible. So I've done the training. You can see how quickly the pixel classifier can detect bright punctate spots based on this simple training via machine learning. Now that I've trained the software, I get this want channel that will be then used in the next step. You notice that with the pixel classifier, the objects in this instance, I haven't spent a lot of time to train the pixel classifier to make the punctate objects as discrete objects, but that actually does not matter in this case because we're going to feed this into the cell analysis recipe that can actually handle that downstream. So right now, I'm actually putting cell input as the green channel, the nucleus as the blue, then vesicle channel as the newly created channel called want. Now I'm going to run a quick cell detection preview. In this case, it's a very much similar interface as what you've seen in the past. You can actually specify the cell diameter, typical cell diameter, which you can measure using the annotation tool. Then you can input that. You can choose to remove the background or add smoothing, as I've shown you in the past. In this case, I've actually worked on the parameters so that you can see that you could detect the boundaries of the cells. Then now I'll move on to the nucleus detection step where I am detecting the nuclei. looking pretty good there. Then I move on to the last vesicle detection step. Now this is the step where I fed the pixel classification step to feed in the channel, and you can see in this example that those vesicles are actually separated into different discrete objects. So you could do per object analysis and you can get per object measurements. Now I've run the entire recipe. You could see that the objects are detected in this example. I'm now going to color code cell membrane by name so that you could see the different cells in different colors. Adjusting the transparency so you could see through. And you can also, in this case, create a workflow. As you saw here, by right-clicking on the object itself, you can create a newly created, our new feature. That's the workflow processor within AVIA 11. What this workflow processor does is it's an enhanced batch processor that actually enables you to input your experimental conditions that you've just created in the step where I've inputted the drug condition, for example, saved as an experiment file format. You can input that along with the condition that was used to create those detected objects into this batch workflow. You can then save that as a workflow file. You can actually add different images, reuse that workflow file, add different experimental files. You could actually sequentially chain a number of different workflows so that one runs after the other. In this example, I'll show you. I'm actually inputting a tag filter so that I only run the workflow on the 4 wells that I've specified. In this example, you're going to recognize that there's a number of different options for export functionality. There's the option to export into Excel. There's an option to export into the CSV file format. That second format is quite important downstream if you want to export it out to another analysis software that you may interface with. In our example today, I'll be using Cytomap as an example to perform advanced analytics. So I'll choose a CSV function there. Moving on to the editor. This is where you could put your notes about the experimental conditions. In this case, I'm changing the name of the workflow file. I am changing the name of each analysis as a field of view. I'm also inputting my name there. This is actually a step where you input a number of different conditions that went into doing your analysis. By creating the workflow, by right-clicking on the object, it's auto-filled in for you, which is a very nice feature. In this case, we're using the pixel classifier I've shown you as the pre-processing step. Then using a recipe I built, which was the 2D cell analysis recipe, and all the other information is auto-filled by creating this workflow. There's also an option for you to do object classification through this step. And essentially the idea is you can chain together multiple different options, analysis options within AVIA, within this workflow processor. Now that I've specified all my options, I've clicked on the run option to run this analysis. You can see that there's a progress bar down below. Each plate in our current setup takes maybe 10 seconds or less, and you can actually view the progress in the queue option and look at the history of detected objects. So this window is actually showing you all the detected objects in the image. Now I can see that the analysis is done, and I can actually open up the newly created experiment file that has all the detected objects displayed on the multi-well setup. So in this case, I'm clicking on the first well, A1, first image, and seeing that the analysis has detected all my desired objects, the cytoplasm, the nucleus, and the vesicles. And you could see when we just look at the vesicles in the no agonist condition, there are a lot of vesicles. And you can go to the spreadsheet function to look at different measurements. Right now we're looking at cell membrane measurements. Now we can move on to vesicles, get individual data points per object. Now I'm going to choose a well. You could spot check this way. You could actually look into a well, look at the first field of view here in the agonist condition. And when we turn off all the other channels except for the vesicles, let's see, I'm turning off the green and the blue. Actually, I want the green back, and turn down the lookup table so that I can actually see. You could see that in this agonist condition, there are far fewer number of detected vesicles. Now we're looking at the newly created option to export into a combined CSV file that you see here. So you see this Excel icon that actually enables you to export all your experiment data per condition you specified per object, creating this mega CSV file that combines all the data in individual object type per condition so that you can actually do downstream analysis. So the way it works is you click on it and you click on the location of the analysis that just was outputted, and this button aggregates all your output into a single CSV file per object per condition. Now you can, in our case, as I mentioned, I'm using Cytomap to showcase how to do downstream analysis. You can search for Cytomap, and I will specify the location of the file where all my combined CSV files are located. Then I'm specifying the X axis is along the centroid X. Then I specify the Y axis as centroid Y. Then Cytomap will import all the data from all the detected objects in AVIA into this spreadsheet.
Once you load the data, you could choose to replace the not available numbers with zeros, which I've done here as the types of analysis I'd like to work on down the road do not allow for zeros, and load. Once the data are loaded, you could choose any of the options on the left. In this first example for Cytomap, I've loaded all the data for both no agonist and agonist groups for all detected cytoplasmic objects and measurements of vesicles inside of cytoplasm. I'll be plotting a simple scatter plot in this case using the new figure button. In this plot, you could choose measurements along which you want to compare your groups and gate your sample for a specific phenotype that you want to further analyze. In this example, I'll choose area, which is the area of each cytoplasm in the x-axis, and contain vesicle count per cell on the y-axis. I'm then choosing to color code the two drug conditions by inputting drug along the z-axis, which is for color. Once you plot this data, you can see that there are interesting group differences emerging along the contained vesicle count axis, as we have already seen in AVIA by visually inspecting detected vesicles. Now, where Cytomap enhances that finding is when you can gate for a specific phenotype of cells by using either the rectangle or polygon gating tools. You could select a subpopulation of cells using this gating tool, from which you could do subsequent analysis based on a specific phenotype of cells you're interested in. Next, I'll be showing you how to run a cellular heat map analysis by going to the extensions menu. As expected, we see a diverging finding on the contained vesicle count dimension. However, we have another interesting finding here where there appears to be another difference along the mean vesicle distance to the center of the cell in these two groups. In this example, I've chosen only a small number of measurements to illustrate the intended workflow. However, when you are working with many different types of measurements, this tool can help you to narrow down which measurements are the interesting dimensions to go after. Lastly, I'll show you a dimensionality reduction tool in Cytomap and how you could use this with measurements from AVIA. Dimensionality reduction is a rapidly emerging tool in the field of spatial biology. In this example, I've chosen a multi-well image set where the absolute XYZ coordinates may not be sensible as measurements. However, there are interesting relative distance measurements such as the mean vesicle distance to the nuclear membrane from AVIA that we can explore. In this example, I've chosen UMAP, the nonlinear dimensionality reduction technique that I discussed earlier, but you could choose from other techniques such as principal component analysis or t-SNE. After selecting from the options, you can see how this tool can help to reduce many number of parameters down to 2 for visualization and data interpretation. Here, I'm inputting parameters for the UMAP technique. I encourage you to play with these parameters to see which help you to interpret your data better. For instance, the n neighbors numbers can help you focus on local structures with smaller numbers or on the global structure with larger numbers. All the features I've shown you today are also available via AVIAweb, which is an additional option. AVIAweb enables you to harness the power of AI tools in AVIA in the cloud now with permanent storage without the need to invest in any powerful hardware. All you need is an internet browser and internet connection, and you can access AVIA from anywhere regardless of the operating system. I want to give special thank you to the entire AVIA team who has created all the features of AVIA 11 that I have shared with you today. Specifically, I want to thank Hongyu Chang, Chicho Huang, Hideki Sasaki, and Hoyan Lai for Cellpose integration, Casey McBride for the Experiment Explorer implementation, Jürgen Reimann for his significant review on spatial biological tools for us to choose Cytomap, Casey McBride and Cameron Shore for the Cytomap export functionality, and John Stansberry for the workflow processor implementation. I want to also thank the entire QA team with Michael Shea, Aiden Maloney-Bertelli, and Grace Lee for their hard work to ensure that AVIA runs as intended. Tasmia Raman Tumpa for insightful benchmarking, Chris Birnbaum for his work on the AVIA TIFF file formats, Maite McHugh, Quen Tran, Hoyen Lai, and Alex Weiss for all things marketing and the sales and support teams for their valuable feedback. My special personal thanks to Hoyen Lai, who has provided endless expertise and invaluable advice on all things AVIA, Konstantin Kappel for his deep knowledge on Leica product and valuable advice on Scrum, and to Sean McElroy for being the best Scrum Master and software team lead one can ask for. And lastly, to Luciana Lucas for his spark of insight and continued encouragement. Thank you very much.
Thank you, Wanyang, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, so let's get started. Our first question here asks, can the new workflow handle 2D and 3D? Wanyang, it looks like you are still muted there.
Hello, Kaylee. How are you?
Thank you.
Thank you for that question. Based on our initial pilot studies, we are fairly confident we can handle both 2D and 3D applications with our workflow.
Great, thank you. Another question we have here, what is the difference between ML and DL?
Machine learning and deep learning are both forms of AI, and both are fairly smart ways of getting problems solved. The difference may be that machine learning learns by training, whereas deep learning can learn from examples much the way human brains can learn using deep neural network models, as I've shown you here. Both are exploding. AVIA has examples of both in our software, and I think we'll be hard pressed in 10 years to find scientific applications that don't benefit from some form of AI.
Great, thank you. Next question here, what about 4D workflow?
based on our current implementation, we are fairly certain that we can handle various types of 4D workflow as well, because we're basically chaining known algorithms and recipes that work in AVIA, and John Stansberry has worked very hard to make this workflow to handle as many applications as possible.
Great. Next question here. Can it be used on processed or unprocessed tissues that are not strained?
I don't understand. At least to me.
It would be great if we can get some more clarifying information on the questions. Would that be more for bright field images, tissue sections, cells? Because we can certainly handle unstained DIC or phase images, no problem. It's built into AVIA.
Great, thank you. All right, another question here. Are all the features presented today available to all AVIA subscribers?
Yes, we're pleased to offer every single feature I've presented here to any AVIA subscriber who is in maintenance. So we wanted to ensure that the benefits of AVIA 11 will be available to anybody who's using AVIA today.
Great, thank you. And it looks like you were able to answer that previous question from that last attendee there, so that's great. All right. Okay, so it looks like we have time. That was our last question here. So thank you again, Wan Yung, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.